Hello, Year 11s. In this lesson, we are going to focus on topic 3 of genetics on a triple award subtopic called protein synthesis. Now, why are proteins so important? Well, actually, proteins make up a whole lot of our body, including our muscles, including our hair, our nails, our enzymes, our antibodies. They are a huge part of our body. Now, as the name implies, we've heard the word synthesis before in protein synthesis. Synthesis means to make. So this is all about how are proteins made. Now, to start off with, our starting point is our DNA, because we talk about DNA being a code, but what we don't tell you is that it's actually a code for the proteins that make us who we are. And this process is a little bit complicated. It's a little bit like my voice has got to be coded into typing, and then that typing has got to be translated into another language. So the first part of this process is called transcription. And let's see why it is called that. So to start off with, I have my DNA molecule. Now, if you remember, your DNA molecule is a double strand, a double helix that is joined together by hydrogen bonds between the nitrogen groups. So I've got T joining with its complementary base pair A with T, C always joins with G, and so on. So I've got my double helix strand, and then along comes an enzyme called RNA polymerase. Now, this is a bit of a naughty enzyme because what it does is it unzips the double strand so that I end up with two single strands. And what it does is it actually binds to the non-coding part that is right at the front of the gene. So it really starts at the beginning of the gene, the section of the DNA that codes for a specific characteristic. There you can see the enzyme RNA polymerase. Now what it's going to do is it breaks the hydrogen bonds between the two strands. And that means that I get left with the strands that are separated. So here you can see is the one strand, ATG, GGA, and so on. Now hanging around in the nucleus are some free nucleotides. Now these nucleotides are made up of RNA rather than DNA. If you remember, DNA stands for deoxyribose nucleic acid because the type of sugar it had was called a deoxyribose sugar. In RNA, it's exactly the same as DNA, except the sugar this time is called ribose sugar rather than deoxyribose sugar. There's one other difference I'm going to show you in a moment. So here we've got all the free nucleotides. Rather than being joined together, they're just loose nucleotides in the nucleus. What do you notice? What's missing? Aha, there are no T's. You're quite right. There are no T's in RNA language. Instead, there are U's for uracil. So what happens? The A from the DNA forms a complementary base pair with the U on the RNA, and that's the only difference there. So when these form complementary base pairs, this is what I'm going to get. The A bonds with U, the T bonds with the A, the G with the C, the G with the C, G with the C, A with the U. Do you get the idea? Quite simple, isn't it? Well done. Okay. Right, so now what we've done is we've successfully changed DNA code into RNA code, and we call the strand messenger RNA because it's not the final product yet. We haven't got protein yet, but it's going to carry the message from the DNA. And what it does is the mRNA strand, the messenger RNA strand, moves out of the nucleus into the cytoplasm of the cell. And this is where stage 2 takes place, and this is called translation. And what we're translating, just like we translate from English to French, we are translating from RNA language into protein language in the form of amino acids. You might remember from earlier on us talking about amino acids being the building blocks of proteins, a bit like me having a pearl necklace like Marge Simpson. The individual beads that make up that necklace with proteins are called amino acids. So let's see what happens here. Well, if you remember, we've always spoken about the place in the cell where protein synthesis takes place is the ribosome for protein synthesis. That's right. So this is where we're going to focus on. Here is my ribosome. It's made out of 2 little parts, and my mRNA strand is going to move along it a little bit like a conveyor belt. And you can see at the moment there are 6 letters or 2 triplets. We actually read it in 3s, which we call triplets. Now, for that triplet, we have special molecules called tRNA, transfer RNA, and they look like this. The transfer RNA carries a particular amino acid, and that transfer RNA matches the UAG. So actually on that tRNA, which I haven't drawn in, there will be for the U, there will be an A. For the A, there will be a U. And for a G, there will be a C. So it's almost got 3 prongs like a fork that matches the first 3 letters. And attached to that tRNA is that amazing amino acid. And you can see for the next triplet, we got GGU. So the code on the tRNA will be CC, and for the U, it will be an A, CCA. And with the CCA tRNA, there is a yellow amino acid. Now, it can only code for 2 at a time. So once that's done, the strand moves along and a peptide bond forms between the 2 amino acids. A peptide bond forms between the 2 amino acids. So now they move along and they wait for the next tRNA to join on, and so it continues. And eventually, I get my protein strand that you can see over here. First had the turquoise, then the yellow, then the orange, and so on. Mission accomplished. I've made my protein. Now, proteins differ by the order of the amino acids going in. And interestingly, there's only 20 different types of amino acids. So the order in which it goes, according to the original DNA code, is critical in determining the type of protein that that particular gene, that section of the DNA, codes for. Now, what I would like you to do, because this is a lot to take in, I want you to read through your notes in your knowledge organizer. And then what I want you to do, using your board pens on the desks, which you will rub out afterwards, I want you to draw out this process with a partner so that you can explain it, and then we will try and find some exam questions on this. Remember, the more you practice, the better you get.